I'd like to first thank the organizers for having me here, and I should be able to share some of my or our research in the field of colon cancer. I have sore throat too, but I have dosed myself with Advil and other medications, so you will have to put up with me. So basically, we have been talking about this for the last 2 or 3 days about various types of cancer and how some of the cancers are on the rise. So colon cancer is the 4th most common cancer worldwide and second leading cause of cancer deaths in North America. Global incidence rates are rising, and it seems to be that in some countries, which are low-risk countries, their incidence of colon cancer is increasing. I will skip some slides because I want to save time. So really, when we talk about colon cancer or cancer prevention, we know that there is a number of factors. They affect the whole process. And they are listed here, and there are probably others which we will come, with time we will find more. In terms of cancer control and prevention strategy, really is to retard, regress, inhibit, or eliminate precursor lesions of cancer in general population or in high-risk population. I mean, that's the idea. And we heard in this meeting early detection, early diagnosis, and that's basically is the idea behind cancer prevention. When we think about how we take an integrated approach for cancer prevention, it's very clear, it's very clear that, epidemiological studies generally generate hypotheses. Sometimes hypotheses are generated based on some of our imagination as well. But basically, they generate hypotheses. In vitro studies, we use them to look at more mechanistic approach to understand cancer. But our interest has been how we can use animal model to really look at the disease process in vivo and how we can really use it in such a way that it can be used for prevention strategy. So in terms of colon cancer, this is just a cartoon of the colon. And you can see that there is ascending colon, descending colon, a transverse colon, descending colon, and then rectum. And it seemed to be that the type of tumors, they appear in different regions are different. And in North America, most of the tumors, they appear in the distal colon. And in low-risk countries like India, Japan, and so on, most of the tumors, they appear in the proximal region of the colon. And what is very interesting is that in North American population now, the proximal colonic tumors is increasing. And we don't know why that is happening. So I left a blank. So I wanted to show you this here about what is the histology of colon. And what I would like you to focus on is the top layer, which is really the mucosa, and it consists of test tube-like glands. Keeping track of that. And generally, when there is changes in the colonic mucosa due to transformation of cells or initiated cells, the glands, they change, and they convert themselves into various types of lesion, adenomas and adenocarcinoma. So you heard this today and last 2 days that carcinogenesis is not a single disease. It goes through many processes, and many, many molecules are involved. So my journey in the field of colon carcinogenesis. When I did my PhD, I was in nutrition and toxicology field. Cancer was not my field. And my first employment was with Ludwig Institute for Cancer Research in Toronto. And they asked me to look for initiators and promoters of colon cancer in our environment, mainly diet. And the model I was given, and this is critical of my own work, the model I was given was to look at, in animal model, we will gavage, extract from food and so on, and look for cell death in the colon. That was our marker. And as a biologist and toxicologist, I started questioning, what is the use of this? It's not specific to colon cancer. So that's how my journey began. And I started looking for earliest changes in the colon, which is related to neoplastic changes. So really, I wanted to develop an animal model which will allow investigation into the stepwise process of colon cancer, and I felt it was very essential then. And we need to, in animal model, we need to mimic low and high risk conditions. And as you know, the seed and soil hypothesis that basically initiated cells will only grow when the environment is right for them. And I'm not going to go into details about why we need to use animal models. I think it's obvious, and I indicated already. Our group became very committed to, my group was really small. I was a young investigator. I had 1 or 2 graduate students. So we became very committed to develop that animal model. I did not want to work with animal model and look for things which are just very transient in nature. So normally when one studies tumorigenesis in an animal model, you inject them with carcinogen and you do intervention, whatever you want to do, and look at tumor incidence. However, if you have hundreds of different compounds or mixtures, you can't do these studies. They're very expensive, and it involves a lot of animals. And this one is the model which eventually we developed, and that is high-risk model where you allow the preneoplastic changes to grow, and then do intervention by diet or any other drug or therapeutic agents. So in 1987, I know it sounds so archaic, 1987, I published the first paper in a journal which had the impact value of 0. And I published this paper where we said I found a change in the colon, the earliest precancerous changes. And the way I did it was totally imagination. I thought if the glands, they change in architecture because of changes of cells and so on growing in it, I should be able to see them. And so what I did is one day I took the colon and I flooded it with methylene blue. The animals were injected with carcinogen. And I was really surprised to see that actually they were there. And here is the picture. What you are seeing here is these circles here. These are the normal crypt. And these are the altered crypts. And at that time, and then when I sectioned these, this is whole mount of colon. It's not histological section. And then I start seeing that actually they grow with time. And you can see them. They are going into, some stay at one crypt stage, but many of them, they grow with time. So you can really look at them with time to see what is happening when you intervene their growth by initiators and promoters. They appear in the colon within 2 weeks, and within 4 to 6 weeks, you can assess the various compounds. This model is now being used by, and you can, we have, and these lesions, they start showing histological changes, mucinous changes very early on, very early on. And now our group and many other groups are using this as a tool to assess. You know, we were talking about Ayurvedic and herbal medicine and so on. They're using this model to look at whether or not they are preventive in nature. And what we have been able to show, and others, that these are putative colonic preneoplastic lesions, and they are induced by many different carcinogens. They are not just one carcinogen. They evolve from one altered colonic crypt, and their growth features is correct. Once they grow, you can count the crypt in the colon, you know, how fast they are growing. And basically, number of things they have. We showed that they had KRAS mutation in these early lesions. And then other groups have shown that there is a lot of biological heterogeneity in these lesions, which you would expect if they are preneoplastic lesions. And basically, this model is now being used to look at various cancer preventive agents. And what is most important is that these lesions are also found in human colon. And so, and there has been association between ACF, I call them aberrant kyphosi, and the risk of colon cancer in human population. So there is validity to use this preclinical model. I'll give you a couple of examples very quickly. I have 4 minutes. Which shows how we are using this. We are also looking at molecular mechanism, which I won't have time to talk about, but I'll give you a couple of examples. So there is some interest in dietary calcium and cancer prevention, especially in high-risk people. So what we did is we mimicked a high-risk model, and we gave different levels of calcium to animal model. So we injected, allowed the lesions to grow, and then we intervene by different dietary calcium. And I know it's a busy slide, but what I would like you to look at is this is the low calcium diets here. 0.1 is low, 0.5 is normal, 1 and 2% calcium is high calcium diet. When you walk on the colon, remember I mentioned colon being distal and proximal and so on. When you walk through the colon, what we found is that the distal colon had an advanced, we looked at advanced ACF, distal colon seemed to respond to high calcium diet. You can see the number of ACF goes down, whereas the proximal colon behaved totally differently. The number of advanced ACF went up. When we looked at the total tumor outcome, what was very interesting is that when we looked at the total tumor outcome in the group, there was no difference. But when we looked at, and here I have just selected 2 regions of the colon, the distal colon and the proximal colon, and these are the number of tumors in the bracket and the size, you can see that distal tumors, they were smaller, and there were fewer in the group, whereas in the proximal colon, the tumors were larger and they were bigger. So ACF sort of mimicked what was happening in the tumor. So in terms of tumor outcome. So the second example is statin and colon cancer. You may have heard people who are on statin, they have lower risk of colon cancer. So basically we try to mimic that situation, and this is a complicated experimental design, but basically what we did is we fed animals 2 types of fat. They're both promoting. One is highly saturated, the other one is unsaturated. And then we gave them lovastatin by gavage each day, only for 3 weeks, because it's a very expensive drug and was very labor intensive. So we said, well, let's see what happens in the last 3 weeks of intervention. And what we were able to show really, I will skip the ACF data, but what we were able to show is that really lovastatin in 3 weeks was able to reduce the tumor outcome in highly unsaturated environment, but not in the saturated fat environment. And also, when you look at the regional differences, most of the tumor reduction occurred in the distal colon. So the proximal colon is behaving differently in response to various agents. And tumor size, most of the tumor which were reduced in this group, unsaturated group, they were smaller tumors. So the larger tumors were able to resist the effect of statin. I wouldn't go through the third one, but in conclusion, I'm running out of time. We have developed a model system which allows really quantitative assessment of preneoplastic changes leading to tumor, and it also allows to screen various cancer preventive agent. And there's a number of ways we can manipulate or exploit the animal model to understand more how we can do intervention in human population which will be effective. And many of you know, many of the intervention trials, which relates to dietary constituents like calcium, fiber, and so on in human population with high risk was not effective. So we need to look at the biology of the disease process, and animal model can be very useful. Thank you.